Hi, I'm Kat,
And I'm Dan.
and we are scientists from the Wilson Laboratories at the Nuffield Department of Population Health, University of Oxford. This talk is about the tests that have been developed around COVID-19, some of which we have been involved with.
We start with a brief introduction to the virus that causes COVID-19 and the impact of the disease. We then explain why testing is an important approach in controlling the spread of the disease and describe the testing methods available, including how we test for current and past infection.
The virus that causes COVID-19 comes from a large family of viruses that can cause mild illness, like the common cold, or more severe respiratory illnesses, like SARS and MERS, which can be fatal. They are called coronaviruses because they have crown-like spikes on their surface, and the Latin word for crown is coronam. SARS was first identified in 2003 in China. It spread to North and South America, Europe, and Asia before the outbreak was contained. Approximately 8,000 people worldwide became sick, and it is believed to have caused 1% of deaths in those becoming ill. MERS was first identified in 2012 in Saudi Arabia. It spread to 27 countries before the outbreak was contained. Approximately 2,500 people worldwide have become sick with MERS, and it's suspected to have caused 34% of deaths in those becoming ill. COVID-19 is the latest major disease to have developed from this family of viruses.
The virus responsible for COVID-19 is called Severe Acute Respiratory Syndrome Coronavirus 2, shortened to SARS-CoV-2. The virus was first identified in December 2019 in Wuhan, China. It has spread to become a global pandemic, and work is still being done to contain the outbreak. It's a huge challenge for scientists, public health officials, and governments all over the world to control the spread of the virus.
To help governments understand and control the spread of COVID-19, scientists and health officials have developed testing programs to help answer two important questions. Firstly, is the person infected with the virus now? In the early stages of the pandemic, anybody with symptoms of COVID-19 were urged to get tested. We now know that the majority of people carrying the virus show no symptoms, and so we also have symptom-free testing available. It is important to identify people who are carrying active virus so they can isolate and prevent others from becoming infected. The second question is, has the person been infected in the past? This may seem a strange question to ask when a person may have recovered from the disease, but knowing information about past infection enables scientists to understand and model how the disease has spread within populations, which may allow further control measures to stop the spread of disease.
There are several ways that we can test for the presence of the virus that causes COVID-19. In this talk, we'll focus on the laboratory-based PCR testing and share with you our experience of setting up a mega testing lab as part of the UK government's response to the pandemic. We'll also talk about antigen testing, which is another method of testing for current infection. We'll then talk about antibody testing, which is how we identify if someone's been infected in the past. And finally, we'll briefly talk about genomic sequencing, which is the method used to identify variants of the virus. These variants may be more dangerous. For example, new variants may allow the virus to pass more easily between people or cause more serious illness. We'll explore each of these testing methods in turn, and be warned, the names can sound a bit complicated.
To begin, PCR testing is what most people will have been familiar with in the early stages of the pandemic. We'll explain exactly what PCR testing is in a moment. People may be tested in hospital, at a regional test site or a drive-in, or receive a test kit at home. A throat and nose swab is taken and sent to a lab for testing. However, in March 2020, at the start of the pandemic, laboratories available for testing were limited, overstretched, and the demand for testing was rapidly increasing. To support the national testing programme, the UK government took the step to develop national Lighthouse laboratories, purpose-built high-throughput PCR testing facilities.
The first and largest of the Lighthouse laboratories was established at Milton, Keynes in the UK Biocentre. We were part of a small team of scientists from the Wilson Laboratories at the Nuffield Department of Population Health, University of Oxford, who were invited to help set up the lab. It was a big job, a large warehouse storage area was being converted into a diagnostic laboratory. We used our experience in large-scale testing and quality systems to set up and validate the laboratory method and put in place procedures to continually check the accuracy of the testing process. We also coordinated the organisation and management of the laboratory and training of staff.
Within a few weeks, the empty warehouse was transformed into a large mega laboratory. A testing process was established within days, and much of the equipment borrowed from university and industry research laboratories. Approximately 200 volunteers with scientific backgrounds were recruited from UK universities and industry. Within 1 month, the laboratory was working 24 hours a day, 7 days a week, and within 2 months, 1 million test results had been reported. A year later, and over 10 million results have been reported by the Milton Keynes Lighthouse laboratory alone.
To operate at this scale, with tens of thousands of samples arriving every day from regional testing sites across the UK, along with home testing kits, we required considerable logistic support. There were so many samples that a marquee had to be erected outside the lab to receive them, and the British Army called in to provide muscle and logistical support to help organise and move not only the samples, but all the laboratory supplies that we needed. All of this effort was to support PCR testing on an unprecedented level, covering the entire country, a feat never previously attempted.
Let's now look at PCR in a bit more detail. Remember, PCR testing detects if a person is currently infected and likely to infect someone else. PCR stands for Polymerase Chain reaction. It was originally developed in the 1980s and is commonly used in research labs. The PCR test is performed to amplify genetic material from a specific organism, such as a virus. If a specific genetic sequence is present in a sample, the polymerase chain reaction will make billions of copies of it, which scientists can detect to determine if someone is infected. SARS-CoV-2 is what is known as an RNA virus. It has single-stranded RNA as its genetic material rather than DNA. The PCR reaction works on double-stranded DNA, so we have to use a type of PCR known as reverse transcriptase PCR, which uses the enzyme reverse transcriptase to convert the RNA into double-stranded DNA. Let's take a quick look at how that works.
Okay, the first step is to extract the RNA from the sample and add a primer, a small manufactured fragment of sequence which complements a specific sequence of the viral RNA. Next, we add the enzyme, reverse transcriptase, and lots of nucleotides, which are the building blocks of DNA. Reverse transcriptase binds to the primer and creates double-stranded DNA by assembling the nucleotides. This part of the process is called reverse transcription. And once complete, we have the double-stranded DNA we need for the polymerase chain reaction.
So now we have double-stranded DNA, we can proceed with PCR. In a PCR reaction, we have a mix of double-stranded DNA, a pair of primers, nucleotides, and an enzyme called polymerase.
The first step in the PCR reaction is to heat the sample to 94 degrees Celsius, which separates the two strands of DNA. This process is called denaturing. The next step is called the annealing step. The sample is cooled to around 55 degrees Celsius, which allows the pair of primers to bind to a complement sequence on the separated strands of DNA. The separated strands of DNA read in opposite directions, which is why you need a pair of primers, one for each strand, to read in opposite directions. Once the primers have annealed, the samples are heated to 72 degrees Celsius, which allows the polymerase to move along each primer in the direction shown by the arrows, and to add nucleotides in what is known as the elongation step.
And that's it. We've replicated the target DNA segment, which can be used as a template for the next cycle of PCR. We keep repeating the PCR process, so after one cycle we have 2 copies of double-stranded DNA. After 2 cycles we have 4 copies. After 3 cycles we have 8 copies, and so on. After 32 cycles we will have over 1 billion copies of the DNA target. And we can do this in under 30 minutes.
In fact, more than one DNA target can be amplified in a sample by adding different pairs of primers. To detect the virus, we actually amplify 3 DNA targets just to test if someone is infected. Each primer has its own dye molecule attached, which can be detected as a light signal each time the DNA target is copied. The more copies of the DNA target, the brighter the signal. Let's have a look at a real test result.
So this is what an individual test result looks like for someone who is positive for COVID-19. The bottom x-axis shows the PCR cycle number. The vertical y-axis shows the light signal intensity of the dyes from each DNA target. Each colour represents a different DNA target. ORF1AB, N gene and S gene are the virus targets. The red MS2 target is an internal control to ensure the PCR reaction has worked properly. For this individual, all 3 virus target genes and the MS2 control have amplified. The colour intensity increases with the number of cycles. This confirms that the PCR reaction has worked correctly and that the individual is COVID positive.
Here is a test result from an individual who is COVID negative. As you can see, the MS2 control is amplified, so we know the PCR reaction has worked correctly, but none of the virus target genes have amplified. That's enough of the theory. Let's look at what actually happens in the Lighthouse laboratory in Milton, Keynes, and go through the steps required to perform the PCR test.
The first step is receiving swab samples which are collected at regional testing sites or from home testing kits. These are swabs from someone's nose and the back of their throat. Lorries can deliver these samples day and night to the marquee, and they arrive in these steel cages. Each swab is stored in a tube, which is sealed in a leak-proof bag. The samples are unpacked from the steel cages and stored into 2 workflows, as some will be processed manually and others using automated robots.
We don't know which samples may have virus present, so every tube is handled as if it's infectious. The next step is to inactivate the virus. A lab operator wearing personal protective equipment, PPE, checks the sample hasn't leaked and removes the sample tube from its packaging inside a biosafety cabinet. These cabinets draw harmful particles away from the operator, a bit like an extractor fan over a kitchen hob, so that the virus can't escape into the room and potentially infect scientists working in the lab. Samples are processed on plates containing 96 wells. Each well is like a tiny test tube. Over 90 samples plus control samples can be tested side by side, and several thousand plates can be processed in a day.
Each sample is mixed with a liquid called a lysis buffer, which inactivates the virus within 15 minutes so that it's no longer infectious. This protects the laboratory scientists from catching the virus and keeps the RNA stable for PCR testing. As in the top photograph, this process can be performed by manual pipetting with operators working in a biosafety cabinet, or it can be automated using a robot liquid handling system, as shown on the bottom photograph. The automated process is capable of working up to 5 times faster.
Once this is complete, it's time to extract the virus's RNA from the sample. Before we start extraction, this is the point at which we add the MS2 internal control, which tells us if the whole process has worked correctly. To extract the RNA, tiny magnetic beads are added to every test well in the 96 well plate. If RNA is present, it will attach to the magnetic beads due to its electrochemical charge. The test plates are then loaded onto this machine, where magnetic rods plunge into each test well. The rods move the magnetic beads, which have the RNA attached to them, between various chemicals to perform the extraction process. The RNA remains firmly attached to the beads until the rods are plunged into the final extraction liquid. Here, the electrochemical charge is disrupted, which causes the RNA to fall off the beads. RNA extraction takes around 25 minutes. Once complete, we are left with a test plate containing purified RNA.
Now we need to prepare the plate for PCR testing. To do this, we add the primer sets for each of the 3 viral target genes and the MS2 control, along with lots of nucleotides and the all-important enzymes, reverse transcriptase and polymerase, to the purified RNA in the 96 well plate. We also add control samples to each plate. In the plates we will show you we have 2 samples that are known to be COVID positive and 2 samples that are known to be negative. We test control samples on every plate to make sure the PCR reaction has worked correctly.
And now we're ready for PCR. We've gone through the theory of how PCR works, and all of those reactions we discussed takes place in one of these benchtop analyzers, known as a thermocycler. These machines control the reactions by changing the temperature during each cycle of the PCR reaction. They also measure the light intensity produced by the dyes attached to the primers for each target gene. So don't forget, if genes from the virus are present, the light intensity increases as the genes amplify with each cycle of the PCR. Now normally you'd expect to see one or two of these machines in a research laboratory. However, due to the large number of samples needed to be analysed, we had over 50 machines running 24 hours a day. It takes 90 minutes for PCR to reach 40 cycles. Once complete, the data needs to be analysed.
Now let's look at what the results from a single PCR plate can look like on a graph. Remember, there are over 90 individual samples and controls on the plate, and potentially 4 different targets that can be amplified in a sample. If all the samples were negative, the graph will be flat as there would be no amplification of viral genes. However, we can see here that there is lots of amplification, indicating that there are lots of positive samples on this plate. Each sample needs to be analysed individually so the result can be reported, but there is far too much information for the scientists in the lab to go through, especially when several hundred plates are processed every day.
So we use computer software to separate out the individual results and classify if samples are positive or not. This image shows the results of one 96 well plate containing 92 individual samples and 4 control samples. You can quickly identify results that are positive and negative, and also inconclusive results, using the traffic light colour system. These are the controls that we added to the plate. If the positive controls are negative or the negative controls are positive, the plate would need to be repeated, and potentially people may be asked to swab again. All results are carefully checked by a trained biomedical scientist before being released to the National Testing Programme. The National Testing Programme have the contact details for the person being tested and will inform them of their result by email and SMS message.